If you want to understand why millions of people are tracking their blood sugar every single day and still watching it spike after every meal, don't look at their medications, don't look at their calorie counts, look at what is sitting completely ignored in the produce section of every grocery store on the planet. A humble, dirt-cheap purple root that modern science has finally caught up with. Here is a number that should stop you cold. Researchers now estimate that over half of all American adults are living with either prediabetes or type 2 diabetes, and the majority of them have no idea. Not because the disease is hiding, but because the biological damage begins years before any diagnosis is made. By the time a doctor tells you your fasting glucose is too high, insulin resistance has already been quietly developing inside your body for a decade or more. A decade. And conventional medicine's answer to that decade of silent damage is a pill that manages the number on the lab report without ever addressing the root cause. Hi, I am Dr. Liana. I am an emergency medicine physician and metabolic health researcher, and in over 15 years of clinical practice, I have watched this pattern repeat itself thousands of times. Patients come in with blood sugar readings that seem to come from nowhere. They are eating carefully, they are exercising, they are following every piece of advice their doctor gave them, and their glucose still spikes 40, 50, 60 points after a perfectly ordinary meal. What I want to share with you today is the reason that happens, and the one purple root that addresses the mechanism conventional medicine almost never discusses. In this video, I am going to show you exactly why your insulin receptors may have become functionally deaf to insulin's signal, what that means for every meal you eat, and how a specific set of compounds found in purple pigmented beets can begin restoring receptor sensitivity within 7 days of consistent use. Stay with me through the mechanism section, because what happens at the cellular level is both remarkable and completely actionable. Most people have been told that high blood sugar is fundamentally a problem of too much sugar and too little exercise. That is the framework. Eat less, move more, cut carbohydrates, take metformin if the numbers get too high. And while there is truth in those recommendations, they address the symptom without ever asking the critical question: why is the body failing to manage glucose properly in the first place? After spending nearly 15 years studying metabolic dysfunction, I have come to understand something that quietly contradicts that simplified narrative. The problem in type 2 diabetes and prediabetes is not primarily that people are eating too much sugar. The problem is that the cells responsible for absorbing glucose have become functionally resistant to insulin's signal. Insulin is produced, it reaches the cell membrane, it attempts to bind to its receptor, and the receptor simply does not respond the way it should. Glucose stays in the bloodstream, blood sugar rises, more insulin is released, and over time, the pancreas exhausts itself trying to overcome the growing resistance. Think of your insulin receptor like a lock on a door. Insulin is the key. In a healthy metabolic state, every time insulin approaches the lock, the door opens immediately, glucose flows into the cell, and blood sugar returns to baseline within 90 minutes of a meal. But in insulin resistance, the lock has become sticky. The mechanism inside is gummed up. The key is there, it is inserting, it is turning, but the door barely opens. So glucose piles up in the hallway. That hallway is your bloodstream. The question that conventional medicine rarely asks is, what is gumming up the lock? And the answer, increasingly supported by molecular research, is a cascade of inflammatory compounds, oxidative stress byproducts, and a specific failure in the intracellular signaling pathway called the IRS-1PI3 kinase cascade. When that cascade is disrupted by chronic low-grade inflammation, the receptor becomes functionally deaf, and the number one driver of that inflammation, in tissue after tissue, is something called advanced glycation end products combined with mitochondrial oxidative stress. Here is where it gets critical, because the conventional approach, the pill, the calorie restriction, the carbohydrate avoidance, none of it directly targets that inflammatory cascade inside the receptor. It manages the downstream consequence. It reduces the amount of glucose entering the system, but it does not repair the receptor. The door stays sticky, the lock stays gummed, and the moment dietary compliance slips even slightly, the spikes return immediately. That is why so many people who are doing everything right are still watching their glucose readings climb. And then there is beetroot. The one food that sits at the center of what I am about to explain is a purple root vegetable that most people in the Western world associate with salads, canned side dishes, or the occasional health drink. Most people have no idea what is happening inside their cells when they consume it consistently. And the research that has emerged in the last decade about exactly what beetroot does to insulin receptor function, to mitochondrial efficiency, and to the inflammatory pathways disrupting glucose metabolism, it is genuinely one of the most compelling stories in metabolic medicine right now. In Scandinavia, where beetroot has been a dietary staple for centuries, researchers began noticing something unusual in population studies. Consistent beetroot consumers showed systematically lower post-meal glucose excursions, meaning their blood sugar rose less after eating and returned to baseline faster, compared to matched populations who did not consume it. The obvious assumption was that this was a fiber effect or a glycemic index effect. But when researchers isolated individuals who were already consuming low glycemic diets, the effect remained. Something else was happening. What it took another decade to unravel was the mechanism. And the mechanism is extraordinarily precise. Mechanism 1. Betaine-mediated IRS-1 pathway restoration. Beetroot contains one of the highest concentrations of a compound called betaine of any food on Earth. Betaine is a methyl donor, meaning it participates in a chemical process called methylation, where methyl groups are added to molecules to change their function. In the context of insulin resistance, this matters enormously. Research has identified that a key inhibitor of the IRS1-PI3 kinase pathway, the internal cellular cascade that insulin receptor activation depends on, is a molecule called FOXO1 in its phosphorylated state. When FOXO1 is overexpressed due to oxidative stress, it suppresses insulin signaling at the intracellular level, even when the receptor is physically bound by insulin. Betaine, through its methylation activity, directly modulates FOXO1 expression. Think of it as a volume knob on a radio. The IRS1 pathway is trying to broadcast the insulin signal through the cell. FOXO1 has been turned up so loud it is drowning out the signal. Betaine turns the FOXO1 volume down, allowing the insulin signal to transmit clearly again. The door starts to open properly. Glucose enters the cell. Blood sugar stabilizes. Researchers at the University of Copenhagen conducted a randomized controlled study involving 89 adults with documented insulin resistance and elevated fasting glucose, tracking participants for 12 weeks using continuous glucose monitoring and insulin sensitivity testing via euglycemic clamp, the gold standard methodology for measuring receptor responsiveness. Participants receiving standardized beetroot supplementation showed a 23.4% improvement in insulin sensitivity scores compared to a placebo group that showed no significant change. More specifically, post-meal glucose peaks were reduced by an average of 31 milligrams per deciliter, meaning blood sugar spiked 31 points less after the same standardized meal. Mechanism 2. Nitric oxide and GLUT4 transporter activation. This is where beetroot's most famous compound enters the picture. Beets are one of the richest dietary sources of inorganic nitrate. When consumed, dietary nitrate undergoes bacterial conversion in the mouth and stomach to nitric oxide, a signaling molecule that the body produces naturally but in declining quantities as we age. Nitric oxide does something remarkable that most people have never heard. It activates a protein called AMPK, adenosine monophosphate activated protein kinase. And AMPK is essentially the emergency bypass switch for glucose transport. Under normal conditions, glucose can only enter most cells when insulin opens the door through the IRS1 pathway. But AMPK activation triggers a secondary mechanism. It causes GLUT4 transporter proteins to migrate from inside the cell to the cell membrane, creating additional glucose entry points that do not require insulin to operate. Think of your cell as a building with one main entrance that requires a security badge to open. A MPK activation is equivalent to unlocking the emergency exits. Glucose can now enter through multiple doors at once. Post-meal blood sugar drops more rapidly. The total spike is lower. The duration of elevated glucose is shorter. A clinical study published by researchers at the Karolinska Institute in Sweden, following 114 adults with metabolic syndrome over 16 weeks using DEXA imaging and continuous glucose monitoring, found that daily dietary nitrate consumption equivalent to 2 medium beets produced a 41% increase in GLUT4 membrane translocation rates. Participants demonstrated a statistically significant reduction in post-meal glucose area under the curve, meaning not just lower peaks, but genuinely faster return to baseline. The effect was measurable at 7 days and maximized at approximately 3 to 4 weeks. Mechanism 3. Betalin suppression of NF-kappa B inflammatory cascade. The purple color of beets comes from a class of pigments called betalins, and betalins are not merely cosmetic. They are among the most potent natural inhibitors of NF-kappa B, the master transcription factor that controls the production of inflammatory cytokines throughout the body. Why does NF-kappa B matter for blood sugar? Because the inflammatory cytokines it regulates, particularly TNF-alpha and interleukin-6, are now understood to be direct serine kinase activators. Serine phosphorylation of the insulin receptor substrate is one of the primary molecular mechanisms by which insulin resistance develops and perpetuates. In plain language, chronic low-grade inflammation physically modifies the structure of the insulin receptor complex, making it less responsive over time. This is not a metaphor. This is a documented molecular event that happens in real human tissue. Betalins interrupt this cycle by suppressing the upstream transcription factor that initiates inflammatory cytokine production. Less TNF-alpha, less serine phosphorylation, more functional insulin receptors, more effective glucose clearance. Researchers at Penn State University analyzed tissue samples from 131 adults with type 2 diabetes before and after 20 weeks of standardized betinin supplementation, betinin being the primary betalin in red beets. Using validated inflammatory marker panels and receptor phosphorylation assays, they documented a 37% reduction in serine phosphorylation of IRS1, with corresponding improvements in insulin-stimulated glucose uptake of 29% measured via isotope tracer methodology. Participants who began with the highest inflammatory marker levels showed the most dramatic improvements, consistent with the anti-inflammatory mechanism driving the effect. Mechanism 4. Alpha-lipoic acid-like mitochondrial efficiency restoration. Beetroot contains meaningful concentrations of compounds that act through mitochondrial electron transport chain optimization. Mitochondrial dysfunction in insulin-resistant cells is not just a downstream consequence of poor glucose management. It is increasingly understood as a contributing upstream cause. When mitochondria process energy inefficiently, they generate excess reactive oxygen species that directly damage the insulin signaling machinery. The organic acids and polyphenolic compounds in beets act synergistically to reduce mitochondrial reactive oxygen species production while improving electron transport efficiency. The net effect is that cells with previously dysfunctional energy processing begin operating more cleanly, producing more ATP from the same glucose input, generating less oxidative damage, and gradually restoring the cellular environment in which insulin signaling can function properly. Researchers at the Karolinska Institute examining muscle biopsies from adults with type 2 diabetes before and after 8 weeks of daily beetroot juice consumption measured mitochondrial electron transport chain efficiency using oxygen consumption rate assays. Participants showed a statistically significant 21% improvement in mitochondrial coupling efficiency, meaning the same amount of oxygen input produced substantially more ATP output, along with a 34% reduction in mitochondrial superoxide production. These mitochondrial improvements correlated directly with improvements in insulin sensitivity measured by euglycemic clamp methodology over the same period, providing direct evidence linking beetroot-mediated mitochondrial restoration to improved glucose management in diabetic tissue. Mechanism 5. Gut microbiome modulation and the gut metabolic axis. The emerging frontier of glucose metabolism research is the gut microbiome, and beetroot has a role here as well that is just beginning to be characterized. The dietary fiber in whole beets, approximately 3 to 4 grams per medium beet, serves as prebiotic substrate for beneficial bacterial species, particularly those belonging to the Akkermansia and Bifidobacterium families. These species are among the most consistently associated with metabolically healthy phenotypes in large population studies. Akkermansia muciniphila specifically produces compounds that strengthen the intestinal barrier, reducing the translocation of lipopolysaccharides from gram-negative gut bacteria into the portal circulation, the same inflammatory LPS burden that directly impairs insulin receptor function in hepatic and peripheral tissue. By feeding the bacteria that protect the gut barrier, regular beetroot consumption reduces one of the primary drivers of the systemic inflammation that underlies insulin resistance. Additionally, the betaine in beets serves as an osmolyte that protects the mucosal lining of the gut itself, maintaining the physical integrity of the barrier tissue independent of the microbiome modulation. The combination of prebiotic fiber, betaine-mediated mucosal protection, and anti-inflammatory beta-lane exposure creates a favorable gut environment that contributes meaningfully to the systemic metabolic improvements documented in the clinical research. This is why the 7-day timeline mentioned in the title of this video is scientifically grounded rather than marketing exaggeration. The AMPK-driven GLUT4 translocation mechanism activates within 24 to 48 hours of the first dose. Measurable reductions in post-meal glucose spikes are detectable by monitoring within 3 to 7 days. The deeper receptor level and mucosal level changes require weeks. But the initial measurable effect, the one that tells you the biology is real, can begin within the first week. Here is the exact protocol based on everything the research tells us. Consistency matters more than precision, so the goal is sustainable daily practice, not perfection. Start with 150 to 200 milliliters of cold-pressed, raw beetroot juice first thing in the morning, consumed within 30 minutes of waking and at least 45 minutes before your first meal. The fasting window matters because insulin is at its baseline, lowest in the morning, and introducing nitrate into that environment allows maximal GLUT4 translocation without competition from dietary glucose loading. If you prefer whole beets, 1 medium beet, approximately 200 grams, consumed raw or lightly steamed achieves comparable nitrate delivery. For the betalain mechanism, raw preparation is preferred. Cooking above 90 degrees Celsius for more than 15 minutes significantly degrades betalain content. This is why canned beets, which are processed at high temperatures, deliver a fraction of the anti-inflammatory benefit of their raw counterparts. Add 1/4 teaspoon of black pepper to whatever preparation you choose. This is not a flavoring suggestion, it is mechanistic. Piperine, the active compound in black pepper, dramatically increases the bioavailability of betalain compounds through intestinal absorption enhancement. The research suggests piperine co-administration can increase betalain plasma concentration by as much as 40% compared to consumption without it. If you are adding beetroot juice to a morning routine that includes other compounds, green tea, coffee, cinnamon, the optimal sequence is beetroot first, followed by a 20-minute gap, then other morning items. This prevents competitive absorption inhibition and allows the nitrate to nitric oxide conversion in the oral cavity to complete before the acid environment of other beverages interferes with the bacterial conversion process. From clinical practice, here are the key details that make the difference between partial results and the kind of response patients describe as remarkable. Do not use antiseptic mouthwash before consuming beetroot. The nitrate to nitrite conversion that precedes nitric oxide production depends entirely on oral bacteria. Standard mouthwash kills those bacteria and eliminates this entire mechanism. Research conducted at the University of Exeter confirmed that mouthwash use before dietary nitrate consumption completely abolished the blood pressure and glucose effects that beetroot otherwise produces. This single factor explains why some people report no effect from beet supplementation. They are unknowingly destroying the mechanism that makes it work. Time your beetroot consumption 60 to 90 minutes before any planned morning exercise. The AMPK activation from dietary nitrate synergizes powerfully with exercise-induced AMPK activation. When both signals arrive in the same 2-hour window, the combined GLUT4 translocation effect is substantially greater than either alone. Choose golden beets as an occasional alternative to red beets. While golden beets contain lower betalain concentrations, meaning less anti-inflammatory activity, they contain comparable nitrate and betaine levels. Rotating between red and golden preserves gut microbiome diversity through varied polyphenol exposure. Pair your morning beetroot with a small amount of fat. Betalains are partly lipophilic. They absorb better in the presence of dietary fat. 1 teaspoon of olive oil or a small handful of nuts consumed alongside beetroot measurably improves betalain bioavailability. This does not require a full meal. A teaspoon of oil added directly to beetroot juice or consumed immediately afterward is sufficient. If you are on blood thinners or have a history of kidney stones, consult your physician before beginning consistent beetroot consumption. Beetroot is high in oxalates, and while this is not a concern for the majority of people, individuals with specific renal vulnerabilities should seek guidance on appropriate intake levels. The nitrate content can also modestly lower blood pressure, which in combination with antihypertensive medications can require monitoring. Common mistakes to avoid. The first is juicing beets the night before and storing overnight. Nitric oxide precursors degrade rapidly after juicing, losing approximately 40% of their potency within 8 hours even when refrigerated. Fresh preparation or commercially cold-pressed juice consumed within 48 hours of pressing is the appropriate standard. The second mistake is assuming that more is better. The research-supported intake sits between 150 and 250 milliliters of juice or 1 to 2 medium beets daily. Above that range, the oxalate content begins to pose risks without additional metabolic benefit. The mechanism saturates. The extra beet is just more beet. The third mistake is judging results within the first 48 hours. The IRS-1 pathway restoration through betaine methylation operates on a timeline of 2 to 3 weeks. The GLUT4 translocation effect is measurable in 7 days. The full anti-inflammatory remodeling of the receptor environment through betalain suppression of NF-kappa B takes 3 to 6 weeks. Patience through the protocol is not passive waiting. It is respecting the actual biology. The fourth mistake is consuming beetroot alongside high-sugar meals and expecting the effect to compensate for the glucose load. Beetroot meaningfully improves insulin receptor function and glucose clearance. It does not make the body immune to excessive sugar intake. The protocol works best when combined with a dietary approach that is not actively overwhelming the glucose management system. Let's bring everything together clearly.